And my office number, I mean, preferable day is Wednesday, but you can show up whenever you want. If I'm around, not a problem. I love talking to you guys. So, I was asked by these adorable ladies to make a show of hands. Did everyone have access to the quiz? If not, show your hand. So, you didn't. So you were, you know, the only one.
Oh.
No, I did it.
You didn't.
Yeah, I did it. Like I had access. Like, isn't that what you were asking?
If you had access, no, I have show hands the ones that didn't have access or had problems in access. So she's the only one. Okay, good. So regarding the questions on the quiz, anything that you guys want to discuss, or did you felt comfortable with all the information coming out from Elliot Marvitz on the video, or is that something that you guys want to discuss before we start? I gotta say up front that we're gonna sort of discuss in detail pretty much everything that Elliot describes on the video, so there's gonna be plenty of information for you to chew on. And up front, apologize that I didn't upload the PowerPoint. The truth is, I don't know how you guys are gonna react to this material, but promise, as soon as the class is over, I'll put in the 2 PowerPoints for this class and the next class, okay? Because this is a completely different kind of approach to what you guys have been doing until now. So, what is a meristem plan? Anyone had troubles with that? Did everyone got the feeling or got the idea that the meristems are basically the stem cell niche in plants? So you guys are familiar with the concept of stem cell niches, right? Everybody knows what is a stem cell, what it produces? Okay. Effects of auxins in plants. Did you got it? I mean, like, did you get it from the video? I mean, are you aware that auxins are basically, you know, a very important signaling molecule in plants, an hormone that basically modulates growth, differentiation, and development? Is that clear from the video for everybody? Cool. How is auxin transported through plant cells? Did you get that from the video? I mean, can anyone tell me what we think is the major transport mechanism of auxins in plants? No? Yep.
The pin?
The pin transporters, okay? So these are proteins that facilitate the efflux of auxins out of cells, which is basically the answer for question number 4, okay? So I hope that you all got it, that from the videos that, you know, basically what these proteins do is they facilitate the efflux of auxins out of cells, okay? And what is the plant cell? What is the involvement of the plant cell on the movement of auxins in the meristem? And I guess that anyone can answer that, or did anyone get that? Yep.
So auxin in the cell, it's ionized,
Yes.
so that it can't move. So it can only go through the transporter. When it goes into the plant cell wall, there it's acidic, the environment, so it protonates, and then
Right.
once it's protonated, it can actually diffuse through the cell membrane. So it's
Right. That's absolutely correct, and that gives me the opportunity actually to, I'm not sure if you guys are used for me using this as a blackboard. I will switch the slides in a minute, but to make a point. So everybody heard about the sodium pump, right? So everybody knows that the way in which animal cells energize themselves is to a pump, which by that I mean protein that hydrolyzes ATP into ADP and basically pumps sodium out and potassium in, okay? Is everybody familiar with this concept, I hope? Now, the reason why you all learned the sodium pump, and you never probably heard about proton pumps, is because we're animals, and we're anthropomorphic, so everything that concerns us is the most important thing in nature, at least for teaching purposes. Because in fact, the most important pump in nature is not the sodium pump. Sodium pumps are specific of animal cells, which is a small subset of all the cells in nature. In fact, the most important pump that actually occurs in every cell, including animals, including ourselves, is the proton pump. So the proton pump actually works in a rather similar way. It's actually the proteins look pretty much like each other, and what it does, it pumps protons out. It's an ATPase, again, if you look at the structure, it's very similar to the sodium pump, but it energizes the membrane. It basically creates a gradient of an ion which is then used for transport of other molecules, okay, either in or out or against the gradient. Now, proton pumps are the major membrane energizers in all the plant cells, all the bacteria, all the fungi, and actually many animal cells, including, for instance, certain parts of our kidneys. Without the proton pump, they would be dead. The consequence, of course, is because protons get accumulated outside, pH drops. Okay, so the outside of the cell walls of bacteria, fungi, plant cells is acidic, which is a concept that we don't have in animals because in our body, practically, we don't have acidity on the outside of the cells. So your colleague gave me a perfectly good answer, and I'm just adding at this point. Is everything clear for you now? Can we start? Okay, I'd like to pretty much start by telling you the story on how these concepts actually get started, on how we actually got there. And so I'm going to start, if the technology helps. Yeah, we got Darwin from all sides, I hope. So I hope you know this gentleman. He's probably the most important person in the history of biology. And you know it, of course, because of the origin of the species and all the work he did with finches and everything. What's probably not so familiar for you is that he worked as much in plants as he worked in animals. Actually, he pretty much got a reputation and got respectable in many respects by the work he produced in plants. And one of his most important or most interesting books is called The Movement of Plants. He did another one, The Power of the Movement of Plants. Basically, he spent years of his life just observing plants and producing these plots, as you can see here. I mean, these plots are very interesting because what he was doing is just, oh, I cannot use the cursor. Okay. So what is your favorite screen for me to use the laser pointer? This one, of course, that one. So I don't have two laser pointers. This sucks. Okay, so I'll try to change. So I'll start by these ones, and then I'll move to that one. And when your neck hurts, I'll probably give a banana. So what he was doing was basically trying to follow the movement of certain leaves or parts of the plant and doing these schematics of the movement of plants. Basically, pretty much he was observing plants for days in a row to observe movements. And obviously, being an experimentalist, trying to do, very naive but very important observations on these movements. And actually, one of these observations, you can actually place the beginning of plant physiology as a scientific discipline, basically in one of these observations. And this is just another few schemes of other movements that he observed in plants. And I'm going to give you a movie so that you guys know exactly how it looks like. And this is seeds of maize. You throw it to the soil. And the first thing they developed is an organ that we call the coleoptile. And as you can see, I mean, this is speeded up, of course, but these coleoptiles immediately start following light. This is an absolutely fundamental adaptation because that's what they have to do. They have to search for lights so that they start developing the leaves. And now the light is going to be switched off, and this light, this bulb, and they're going to switch off the light of, I guess, the ceiling, and you're going to see what happens to the coleoptiles as soon as that happens, which should be about now. Or now. There you go. And as you can see, I mean, immediately the coleoptiles respond and start drifting in another direction. In other words, they have ways not only of sensing light, but actually be able to reorient their growth towards the light. Okay? And Darwin, I mean, pretty much observed the same response, but he did a few more experiments, and this actually begins the whole story that you've seen from Elliot Marwitz about the auxins, because, I mean, what he observed was that if you would kind of chop the head of the coleoptile, this, terminal part of the coleoptile, then the coleoptiles would lose the response to light. And furthermore, if he kind of made a small hat and put the hat on the top of the coleoptile the same way, I mean, things would not bend. And pretty much he concluded two things. One, the photoreceptors or whatever senses the light, I mean, they didn't use the term photoreceptors in those days. It's placed right at the tip. And secondly, there's got to be something that moves along the coleoptile that actually transports that information. And this is relevant for the context of the class I'm supposed to give you today, which is cell signaling and development. In other words, there are signals that move from certain parts of certain cells in an organ to other parts of the organ and inform the other parts of the organ that something has to be done. And this is absolutely fundamental in terms of cell biology when you get to multicellular organisms. Basically, multicellular organisms have to divide information, have to divide function, but they also have to communicate. So all multicellular organisms have some sort of cell signaling mechanism to coordinate development and to pattern development. In other words, a cell becomes differentiated when a certain number of mechanisms or signaling mechanisms tell them you got to be different, or in this case, grow more, so that the coleoptile bends. Now, other people followed up on these experiments. I mean, you see a gap here of more than 30 years. Actually, this is not real. I mean, people were doing experiments in this interval. But I mean, the next ones that became famous because they give rise to the discovery of this hormone, the auxin, is actually the experiments of Boyce Jensen. So what they did is they did, again, very naive experiments. They just like kind of put in little pieces of very thin mica barriers. I mean, the idea was if there's something like going down or going up according to one of the sides or the other side, I mean, just putting a physical barrier should stop them to move. And in fact, when they place them on the opposite side of the light, the coleoptile does not bend, but when they put it on the side of the light, the coleoptile bends, which led them to conclude that there's something that moves on that side, on the opposite side of the light, either up or down, we don't know, and that is not basically stopped when they put it on this side. And another experiment that they did actually was to chop the same experiment that Darwin did, but instead of just let it out, put just a gelatin barrier in between, and that does not stop the coleoptile bending response, which basically they concluded there's got to be a substance that goes down, which is diffusible to the gelatin, and therefore if there is diffusible to the gelatin, I mean, they start kind of doing this kind of accumulation of these bits of gelatin to actually try to extract what is the substances that are actually segregated and serve to do the cell-to-cell signaling. I mean, basically, I mean, this led to the discovery of auxins. So this was the first plant hormone described. Of course, there were a few already in animals, but I mean, this was the first one. And to our knowledge, still up until today, it's the most important one. And that's why pretty much, if you want to talk hormones about plant hormones to anyone, I mean, you should start by auxins because they're really the most important one. Basically, I mean, there's a number of other experiments. Either just substituting the tips by blocks of gelatin that were incubated previously with coleoptiles, same sort of effects. So it all pointed out to the fact that there was a diffusible substance that was promoting cell-to-cell signaling and making these cells from the outer part of the coleoptile to elongate more than the cells on the inner part of the coleoptile to produce the curvature. And this is pretty much what was later quantified when the first auxin, I'm gonna kill this guy with a laser, sorry about that, with a laser pointer. When the first auxin called indole acetic acid was basically discovered. And as you can see here, if by just putting certain amounts of this auxin called indole acetic acid, I'm just gonna show you the structure in 2 slides. I mean, basically you can get various bending effects. So this mechanistically said, this is the molecule, indole acetic acid, which is used as an hormone to communicate cell-to-cell signaling, to say that side elongate and the other one not. Okay, auxin, again, the first auxin to be isolated was precisely indole acetic acid. This is what you get when you look at the relative elongation in these coleoptile assays. This is a very important piece of observation. Please bear this in mind whenever you speak about hormones. Most hormones have biphasic effects. So there's an optimal concentration, and to the left and to the right, there's actually, it goes up, it goes down. And from a certain concentration, most hormones, if not all, have actually inhibitory effects. So every hormone, or practically every hormone, works like that. And that's actually a bit of a hallmark of an hormone, to have this kind of biphasic and eventually inhibitory domains of concentration. And actually, when people search for hormones, this is kind of the curve that people want to get, because this is an indication that probably this is a substance which is working as an hormone. We could discuss why this happened, but I mean, if you have curiosity on that, you can talk to me later, but we don't have time to discuss this now. But there's a good reason for this to happen. So it works on very low concentrations, like a good hormone should work. So the rest of the things had to be borrowed from the animal field. Of course, in the animal field, the definition of a hormone also relies on the definition of what is the producing organ and the target organ. And in plants, that is not so easy to apply, because in principle, all plant cells can produce all hormones and can be sensitive to all hormones. So the issue of target and emitter, it's not so well defined in plants. And still we talk about hormones because obviously they're produced by all cells but are specifically activated in certain concentrations in certain tissues. And that's the purpose of these two classes, to understand how that happens. This is the chemical structure of the three most well-known naturally occurring auxins. I mean, the most common one again is the top one, IIA. Indole-3 acetic acid. There's two others that which are known. I mean, if I don't say anything from the rest of the two classes, I'm just going to be referring to the first one because that's the most common, and that's the one that most people use when they're doing experiments. It's derived, I mean, a number of synthetic auxins are known. I just put this here because usually if you go to the literature, the one which is more used for experimental purposes is this one, the naphthalenic acetic acid. And there's a good reason for that. While this one is natural degraded in tissues, I mean, this one is not. The other four, I mean, they're very important, not exactly for, at least for you guys, but for the farmers, for good reasons, because there are insects. In other words, if you put too much of a good thing, a plant eventually dies, which is that part of the inhibition of the curve I just showed you. I mean, these auxins, these synthetic auxins, are so strong that basically if you put them on large amounts inside the plant, I mean, basically the plant just dies because everything gets inhibited by it. And these are actually, this one is very much used, and it's a commercial herbicide. Okay, just you don't have to go through all the steps. I just wanted to point out the thing. I mean, auxin is derived from tryptophan, so it's an amino acid derived hormone. And I just point this out to just let you guys know that it's very common in nature that you find the same building blocks are used by different organisms for completely different things. So tryptophan is also the source of another important animal hormone called serotonin. Many of you guys probably heard of it, and it's very important now because most of the pills you take for depression act precisely on serotonin. And this serotonin actually on the pathway of another important hormone, which is melatonin, which is used for regulating our dark cycles. And the reason why you go to bed at night is because you have a peak at melatonin at the end of the day. So same building blocks, tryptophan, are used for hormones in animals and plants. And you see that over and over, evolution kind of picks up and co-opts building blocks to the different things in different organisms. This is the point here. Just don't forget that auxins come from tryptophan. It's the only thing this slide said for. Okay, so comes the direction of the target tissues and what is the donor tissue and the target tissues. And those experiments, again, were done using coleoptiles or small seedlings. So what people were doing is they were collecting segments of these shoots and basically putting blocks of agar in one of the extremes or in the two extremes and actually trying to follow the direction of where the auxins were going. And if you just put the block on the top part, then soon enough everything is distributed, and that experiment tells you basically that auxin can diffuse very easily from the apical part of a plant to the basal part of the plant. And this is a very important concept because what I'm trying to introduce to you here is that plants know what is up and what is down, okay? For a good reason. Up, there's the sun, needed for photosynthesis. Down, there's the water and the minerals needed for everything else. Okay. Now, if you do the reverse experiment, it looks like, not much goes up. So what can you conclude from this experiment? One answer? Yep.
First one down.
So preferentially, auxins to be translocated on the apical basal direction, okay? And of course, you can do that with other sorts of experiments and eventually do other sorts of experimentation. I mean, but what is the basal conclusion or the basic conclusion of this kind of experiments is that apparently there's a preferential transport of auxins from the top to the bottom of the plant. And of course, this got people thinking about, oh, maybe then there's a mechanism by which plants knows what is up and what is down, based on the transportation of auxins. Okay, and they start doing a number of very interesting and very creative and again very naive experiments that actually show that. Say, for instance, if you pick up a plant and you chop the stem of the plant in little pieces and put it just in vitro, like in agar with some nutrients. I mean, as you know, I mean, plants have this thing called totipotency. I mean, that they can regenerate organs from any cell. So the fact, I mean, if you cut the finger, which is not a very wise thing to do, you don't expect that the finger to grow anywhere. But in plants, I mean, you can cut any part of a plant and you know that it will regenerate. Okay. So what these experiments actually show you is that if you cut a number of segments and you just put them in agar, magically they retain the memory what was up and what was down. And the reason why you know that is that the part in which the new stem and the new roots arise, it's always the one which was up and down respectively. And that is independent of the positioning on the plant, which tells you that the plant has a memory or it has a system that tells them what is up and what is down. Same thing if you do grafting. I mean, everybody knows what is grafting. Picking up a piece of a plant and putting it into another plant. If you graft a piece, say a meristem or a piece of a plant in one of these stem sections, and you follow where do the vascular bundles are formed and where would the roots will be formed, it's always in the direction apical basal. So somehow plants have information that tells them what is up and what is down. And for instance here, I mean, what these lines here represent are actually the vascular bundles. So basically the veins and the arteries of the plants. And they're all pointing out in the direction. They're formed in the direction of the root, because that's where they have to get the water and the minerals, for good reasons again. Now this can even be led to more dramatic things, and these are just a number of surgeries that people have done, namely this person that wrote this book, an Israeli person by the name of Tzisash. And these are a series of incisions he did in stems, and as you can see, I mean, when he makes kind of a simple incision, I mean, the new vascular bundles which get formed, again, follow the route of up-down. But here's when he got creative. He said to himself, is there like a limit to that? Can I revert that in terms of the physiology or the anatomy of the stem? So he started kind of developing more and more intricate and more complicated surgery cuttings on the stems. And as you can see, I mean, at this point, there's no apical basal anymore. I mean, it's perpendicular. So, I mean, there's got points in which you can actually make incisions and you can actually revert the apical basal information. And pretty much, this is a very important observation. Anyone can tell me why this is a very important observation? So, what we are representing here by the single arrow is the direction of the formation of the new vascular bundles, and this big arrow tells you what is the apical basal axis. So this segment here seems to be inverted. So what could this experiment possibly tell us? Yep.
Maybe this direction isn't just because.
Well, yeah, you're already putting in one thing that I didn't call into the board, but you're absolutely correct. You spoke about gravity. You could speak about light, which is in principle the thing that brings them out, but that tells you that, I mean, it's not just where the stimuli comes, but apparently it's how the flow gets channeled, okay? So in other words, what you can conclude from this experiment is what is important is not just the positioning, but the direction in which things are flowing. And this is a very important concept, because then you have to admit that the positional information is not just encoded on the anatomy, but it's encoded on the flow of something. Okay? And you can start asking another question. What is this something? Why is it flowing? Yep?
I'm a little confused. Are we saying that the flow follows the cut, or that it follows the...
What I'm saying is that what follows the cut is the formation of the vascular bundles, and that is following the flow of something that comes from up down. And if that thing is forced to take an S, like in this case, then the vascular bundles will interpret the flow instead of the position. Okay? Okay, long story short, people already knew auxins when these experiments were done, so pretty much they came to the conclusion, and I think there was a slide in Elliot Marovit's talk showing pretty much this scheme, that it might be auxins, okay? Because I've showed you the other experiment with the agar blocks. Auxins go from the top to the bottom, or preferentially go from the top of the plant. So people came up, and this, as you can see, we're talking about information from 1983. So this was the best that people could know in 1983. Some of you were not born, or most of you were not born. Yes, but it's relatively recent if you come to think about it. It's like 30 years ago. It's not that long of a time. So this is what people knew. They knew that cell walls are acidic, and at the pH of 5.5 or around that, where cell walls are supposed to be maintained, indolacetic acid is in the conjugated form. In other words, it doesn't have a charge, and therefore it can cross the membrane without much, just following the gradient, or through some facilitators using proteins. This was known already. Now, this is the cool thing. I don't know if you got that completely from the video, but this is the cool thing, and this is why this scheme is so ingenious and so smart. Once they get into the cell, because the pH in the cytosol is around 7, they get protonated. In other words, deprotonated. In other words, they become charged. In other words, while they go inside with the gradient, they get trapped inside the cell, and therefore they create another gradient. It's the same molecule, but because outside it's non-protonated, it's protonated, and inside it's non-protonated, actually it can move from one side to the other just by gradient. Okay, where is the energy coming for this transport? Why is this possible? Because, I mean, it cannot just go inside and outside without energy. Where is the energy coming from? From the proton pumps. Basically, the energy is coming from these guys here, the proton pumps, and it's encoded in the form of a pH gradient. Okay, so this is where the energy is coming. I mean, you have to think that when a molecule goes from one side to another side, it's got to be energy of some sort. Okay, if not directly, indirectly, and usually, I mean, the sources of energy are always these ATPases. Okay. So the beauty of this model is then you could explain the flow of auxins from the top to the bottom just by admitting that there should be a channel or a protein which would facilitate the sorting of the deprotonated indolacetic acid, IAA. But this is the trick, and this is, look, this is 83. This is the prediction. If such a flux facilitator would occur, and if this is true, that what is important is the flow of auxin, then it should be on the basal part of the cells. This is a very strong prediction. Okay? 1983. So what would be this efflux facilitator? People don't know it. Didn't know in those days. And this is just another scheme, a little bit more complicated with more details, but pretty much it tells you the same. So we know how auxins would get into the cells. We don't know how auxins get out of the cell. And if by any instance this protein that actually helps auxin to get out of the cell is placed on the basal part of the cells of the plants, then we would have ourselves a mechanism of auxins from going from top to down, and all the plant would know what is top and what is down, because that's where the flow of auxin goes. Okay? Now, let's step to 1991. Okay? This paper, I mean, this guy will never probably win the Nobel Prize, of course, but this paper started a small revolution in plant biology. Now, we're talking about a era in which Drosophila, C. elegans, plants, Arabidopsis, people start doing genetic screens. So, are you familiar with the concept of genetic screens? It's intellectually the lowest, poorest method of doing research, but in real life, the one which has been more useful in the past few decades to discover new things. It's like shooting with a blindfold to a tree full of birds. You don't know what you're going to hit or what's going to come down. But you have a faith, because the tree is full of birds, that you shoot at the tree, you're going to get something. Okay? Did you get? This is the best definition that anyone can give you of a genetic screen. Okay? put a blindfold, you have a tree full of birds and shoot, and something will come out. Okay? Then you just have to pick it up and do something with it. So what these guys were doing is this is the beginning of the genetic screens, so any phenotype was good. Now these days phenotypes are becoming more and more and more sophisticated. I mean, the one I like to quote here is a phenotype on the defecation cycle of C. elegans. Yes, C. elegans do poo. Everybody knows what is C. elegans? And they poo with an incredible regularity. They poo every minute, like, okay? So this guy in 2003 made a screening on the pooing cycle of C. elegans and find it. This is a Cell paper. Cell is like the best journal you can get, and found 3 new channels, calcium channels for science. And you say, wow, this is a lot of science just out of pooing. But it gets better because 5 years later, he made another screening in which he's not looking at the poo, but he managed to train some of poor PhD student or postdoc, whatever, to be able to recognize that they just don't poo. There's 3 phases on the pooing. There's like when they contract, and they like poop, and then that's when they kind of relax. Okay. Now we're joking, but it's another Cell paper. Needless to say, this guy probably got millions in grants to continue his research and became very famous. Actually, he died a year after. But he discovered 3 completely new mechanisms of neurotransmission. And he was the first person to actually establish that pH or protons can act as neurotransmitters because he found 2 proton channels on the relaxation phase. Well, okay, so what is the story here? The story is screenings are getting more and more sophisticated, more and more precise, okay? So if somebody tells you, oh, if those of you that want to follow research and, oh, let's start the genetic screen, you better ask what, okay? Because it's getting very, very, very sophisticated. But in those days, anything would go. Let's do a genetic screen and see if there's no flowers. Yeah, let's do that. And people will find things. Of course, it doesn't work anymore because everybody knows what are the genes that make flowers down. So what these guys observed was this mutation kind of in an Arabidopsis, instead of having a shoot full of leaves and flowers, the shoots were naked. No leaves or flowers. Okay, so we have a new class of genes that apparently when they are mutated, or a gene when it's mutated, apparently everything that promotes the organogenesis of flowers and leaves, it's blocked. And he could have stopped there, and he probably would get a very nice publication in those days because any mutant in those days was published, basically. But they went, a mile down, and actually they said, what could prevent the formation of organs? And of course, people knew that auxins were involved in the specification of organs. So they went on to measure auxins in the different tissues, and basically this is the plot that changed everything. So these 3 big bars are wild types or conditions in which they could observe wild type responses, and these ones are various organs in which they measured the auxin levels in the mutated plants. And immediately they concluded, oh, there's a problem in the transport of auxins because these guys have much less auxin levels than the wild type. This plot alone, I can tell it probably took them like years to produce because measuring auxins directly, it's a pain in the neck. As you saw, like any hormone, they occur in very, very low concentrations. So isolating hormones, it's still, a huge endeavor. So we spoke about melatonin, just to give you an example. So melatonin is produced in an organ we have inside our brain, which is called the pineal gland. So the person that isolated melatonin, the gentleman, the chemist, the American chemist by the name of Lerner, had to sacrifice 5,000 pigs, which has the biggest pineal gland that you can think of, kind of the size of a golf ball, to isolate 1 microgram of melatonin, 5,000 animals. Okay, so it's not easy. And then if you see the quantities which are here, we're talking about molecule. Yep.
What about just like melatonin supplements we have today?
They are extracted either from plants, or they are extracted from, or they synthetically derivative. It's a relatively simple procedure from tryptophan. Okay, very good question. No, it's not a bunch of pigs being sacrificed for you to sleep better at night. No. Don't worry, you can continue to take it if that's your fault. Okay, so why is it called pin? Because it looks like a pin. So I mean, like, you know, people are not very creative in giving names to oh, they are. You know, yeah, actually, people are very creative in giving names to mutants, but not in this case. It looks like a pin, so it's a pin. You know, but you know, I mean, you guys are going to med school, I hear. Most of you want to, right? So there was kind of a, well, not this, but there was a bit of a polemics a few years ago because, you know, we research scientists tend to give funny names to mutants, and sometimes people find, you know, very serious diseases that have the same mutation, and then they have to, you know, talk to people. Hey, you have a mutation on a very funny name, and people don't find that very funny. And of course, hedgehog is the most common because it's one of the most important genes in animal development, and of course, it was named after the cartoon character. But my favorite is Ken and Barbie. Anyone knows Ken and Barbie? You know, you know what it?
It's a Drosophila.
Yeah. And what is the phenotype? No genitalia, basically. Turns out that there's a human mutation, so and it's kind of observed when the baby comes out. So I mean, I can imagine that if I was not a doctor and the parents come, yeah, well, we have bad news. Your son has a mutation on Ken and Barbie. I wouldn't find it. So there was a movement of certain medical associations that all the mutants should be renamed to like XPTO1234 or whatever that took all these funny names. Anyway, these are pins because they look like pins, okay? And this is the beginning of the story. So, this paper alone, I mean, must have produced more than 100 papers in Cell, Nature, and Science in the next decade. But this is very recent. I mean, we're talking about 13 years ago. You were already born, okay? I mean, people, of course, went to bioinformatics, and they end up finding that this is a very, you know, there were a number of members in every plant. In Arabidopsis, which are genetic models, there are 8. 7 of them are pretty much expressed in many tissues. Number 8 is only expressed in pollen. I'm not going to go to that, but at least 7, not just 1, could be involved in the transport of auxins if that was the verification that these Japanese authors made. Of course, there was this model that said that we need something which is located on the basal part of the cell. And now this Japanese team says, oh, maybe this is it. So what would you be your first experiment immediately? Like, this is going to be a Nature paper. Join these two facts together. What, yeah. Go.
Sorry.
What would be, you know that there's a model that says there's got to be an efflux facilitator. I mean, if this whole thing works just by admitting that there is a facilitator, a protein that facilitates auxin efflux on the basal part of the cell. And then these guys say, oh, this protein seems to be an efflux transporter. What would be the first experiment you will run to the lab to do?
Knock down the gene and see.
Well, they already done that. Next. To do what? Probe to auxin flow. That would be good, but that only came 10 years after. I'm going to tell you that story. Next. The obvious one. Come on, guys. You know there should be something in a place, and then there's somebody that says, this may be the something. What do you do next? Localization, okay? Check if it's there, okay? Okay, so you guys have been talking about GFP a lot, I suppose. Now, you take it for granted, because everybody's doing it, and you know, any undergrad student can do a GFP experiment these days. I mean, in the next, you only have to go 20 years ago, people didn't have these tools, you know? So people didn't know where proteins were in the cell, had no clue. So just try to interiorize the fact that nobody answered this, the fact that localizing proteins or localizing something in a cell sometimes is the first step to understanding function. Okay? So if you have a model that says this should be here, and somebody says, oh, this might be it, the first thing you're going to see is verify, is this it? Is it in a right location? Okay. And this is absolutely fundamental. When you want to understand what is a gene function or a protein function, one of the first things that you usually do is, where is it? And try to see if that fits with some function that you kind of arbitrate should be important for it. Okay? So what would be the prediction then, if this was it? That it should be on the basal part of the cells, whatever cells they observe. Okay? These days, people didn't have GFP, or it was not so easy to do GFP, so the way that people did it was with antibodies. You isolate the protein, you inject in a rabbit, take the serum out, and then you react with the tissue. And as you can see, the first guy that made this observation, I gotta tell you, he must have pulled out of his socks immediately, because as you can see, the protein is perfectly localized to the apical part of the cell. In this case, not all cells, this was perhaps the surprise, but in these root cortex cells, it's perfectly located in the basal part of the cell. So people, wow, cool, now we have everything. Auxins comes from the apical to the basis, they flow, we know the mechanism, they go inside the cell because they are protonated, they come out of the cell after deprotonations to pin 1, world is perfect, okay? But don't forget there's 7 more pins. And of course, then a stampede started, like the best things in the world in plant biology jumped into this like a pack of wolves, because everybody wanted to have the prize of understanding how this works. Mind the dates. We're talking about 2008. It's not too long ago, okay? Less than 20 years, everything that you're going to see happened in less than 20 years, in the past couple of decades. So this is the prediction. So maybe then, if this is the fact, when we look at the localization of these pin efflux carriers, we can deduce that auxins move in certain movements. And immediately, as you can see in the video from Elliot Meyerowitz, people try to see what happens in the apical meristem. What is the localization of pin 1? It turns out that pin 1 in the apical meristem was localized on the epidermis. So, okay, there's a pattern here that I want you to keep in mind. So in the root, pin 1 is inside the root. In the shoot, pin 1 is on the epidermis. So obviously these things are not expressed just anywhere, they're expressed very specifically. But again, as in the root, is expressed only on one end of the cell, which kind of makes you think, maybe this is a signaling molecule. This is what the cells, use to talk to each other and produce effects. Okay, well, comes pin 2, because people start cloning all these ones. Now we know there's a family that does that. Let's see what the other 7 are doing. It turns out that pin 2 has a slightly different and more complicating, for the model at least, localization. So if you look at the cortex, they're on the basal part of the cells, but if you look at the epidermis, they're on the upper part of the cell. So it looks like we have a highway here. I mean, in the cortical cells, if the model is correct, auxin is going down. In the epidermis, if the model is correct, auxin is going up. Does that make any sense? Can we make sense out of this? And at this point, we have to make a parenthesis. I mean, somebody already said that we should look at the levels of auxin. I think you, right? And I told you, I mean, that these Japanese were really kind of very brave, because they went to actually quantify that, and that's very difficult. You cannot get one of these cells to say, how much auxin do I have inside? It's impossible. You cannot do that, okay? So people start thinking at this point, man, it would be cool if we had a probe, like a fluorescent probe for auxins, because there was none, which could tell us, okay, auxins are very accumulated here or less accumulated there. Now, the way that it was done was incredibly, incredibly clever, and I'm just going to spend a few slides to tell you how it is, because there's a bunch of probes now showing up, even for the people that has a medical inclination, like as clinical probes, which are produced in this way. Okay. Auxins have all these effects that produce growth and development and everything. So people went to look for receptors of auxins. And what they found out was actually that the receptors of auxins, are in the cytosol. Am I getting a signal here? Yeah, there you go. Are in the cytosol. So if this is out and this is in of the cell, and if this is the nucleus. So auxins go inside, they bind to a receptor which is cytosolic, and this receptor migrates to the nucleus and basically binds the DNA to induce the induction of some genes. So this is a scheme which is exactly the same, for instance, as our steroid hormones do. So progesterone, testosterone, they work exactly the same way. They go into the cell to bind the receptor, and then the receptor and the thing goes into the nucleus to do something, which is usually to induce the expression of some genes. Okay? So people found the receptors of auxins, and more than that, and these things were called auxin responsive factors, basically. And these transcription factors basically were found to bind to the promoters of some genes. Is the concept of promoter familiar to everybody? What does the promoter does? Is a promoter part of a gene? No?
I mean, it binds, and then there they know that from that point the transcription starts from the gene.
It depends on how you classify the things, but I would tend to say that it's probably more correct that you call if gene is the hereditary unit of transmissions, then the promoter is part of the gene. Okay?
Okay.
So, I mean, I don't want to be too dogmatic on this, but I think that most of the textbooks and most of the people that work on these things would say that a gene actually includes the promoter. Okay? And so when you clone a gene, I mean, usually you clone also the promoter. And in fact, if you want to, I mean, I'm not being too dogmatic on this, and I accept that other people have other views. Well, what you're saying is that you were only considering the gene what we call the open reading frame, right?
Yes.
Okay? Because that's what encodes the protein. But if you take a cell and developmental perspective, there's information which is as much important in the promoter as in the orphan. Because that's where that's where the DNA sequence is that tells you when and where the gene is going to be expressed. Is everybody following me? Because promoters is like the most important concept in biology in the last 30 years. Okay? And you're going to see an application in a minute that will tell you how this important is this.
So when they have like the human genome sequence and specific genes, they include the nucleotides in the promoter?
I mean, by definition, the promoters are the, I mean, if you want to search for the promoter, by definition, you say that the promoter and the sequences of DNA which are responsible for the expression of that specific orb are 1 to 1.5 kbs upstream of the initiation code. Okay? And yeah, that's what they do. They sequence everything, and they classify that as also, I mean, usually what you get from the database is the ORF, but you know that the 1.5 kb upstream is the promoter. And that's where the information is for the gene to be expressed, which in terms of cell and developmental biology, the information where the genes should be switched on and off is as important as the protein that's coded. Otherwise, if all cells would express the 25,000 genes, instead of us having two ends, and we would just be a big giant meatball rolling over something, because all the genes would be expressed everywhere. Okay, so what makes it a hand and a head is the different promoters are activating different proteins. Is that okay? Okay, promoters bind transcription factors. Is the concept of transcription factor familiar to everybody? At this point, I usually ask people to say with me, promoter is DNA, transcription factor is protein. Can you say that with me? Sure. Can we say everything like chorus, like the promoter is DNA, transcription factor is protein? Because I know that when I go to the exams, if I don't do that, half of you would mix up everything. Okay? So promoter has the information in the DNA. Protein is, transcription factor is the thing that binds to the promoter and switch it on or off. Okay? So ARFs, these auxin responding factors, are basically transcription factors that bind to promoters. So when people found them, and the way they found them, I'm just going to put this slide so that you guys know, is by this DNA's footprint. So what you do, you run a gel, and then you stain the gel with an antibody for DNAs. And when you see a blank, when you see a blank, sorry, forgot about this. When you see a blank, that's probably the region of the DNA where whatever you're testing is bound, because if you have a protein bound to the DNA, then the DNA does not bond to the DNA. Does that make sense to everybody? So it's kind of a negative printing of where you have proteins. And then, of course, because you know the sequence, you can go back to the sequence and see where it binds. So people found that there were promoters that were kind of oxygen sensitive. Okay? Now, and there's a second paper in which people actually picked up these natural promoters where these ARFs bind and actually optimized them. And if you see this bar plot here, these are the natural promoters, the ones that occur, you know, in the plant. And these are promoters that people optimize by changing, you know, one by one the nucleotides to make them much more efficient. Okay? So what did they got here? They got artificial promoters that respond tremendously well to oxidants. True, and let's make the true scheme now. So if you notice here, the one that gives the higher response is this one that they call DR5. Okay? So DR5 is a promoter. So DR5 is in the DNA. So there's DR5 here. Okay? And the way it works is the following. So you have the cell, you know, auxin comes in. Auxin binds to, you know, ARF, auxin responsive factor, goes into the nucleus and binds to DR5. Okay? Now, when you have a promoter, what can you do next with a promoter to make it a reporter?
Put the promoter in front of something that's easy to visualize.
There you go. That's where I say that this is my concept of a gene, because it's a functional concept. If you have a promoter, you can put anything here. It can be your gene, or it can be anything else. So what would be one thing that you would put after the promoter to make it visible? GFP, okay? So, and what would you obtain? Say, like, if you flood a plant with auxins, what would be your expected result? You should see a lot of GFP, and the quantity of the GFP would be an indirect measurement of the quantity of auxin. Is everybody following me? Because many of the kits you're going to use to check if people have certain proteins on the blood are based on these kind of concepts. Okay? I mean, the other one, just the finishing, that people use very often in plants, it's a gene called GUS. Have you guys discussed it, GUS? It's an enzyme which is a bacterial enzyme. Doesn't occur in plants. It's called beta-glucuronidase. And the cool thing is that it produces a very nice, beautiful blue color. So it's an alternative. If you don't like green, this produces blue. I mean, one word of advice. GFP can be used on live material. GUS, you have to kill the material to see it. So that's the major difference. GUS is a little bit more sensitive, but you have to kill the material. Anyway, that was the first one that people used. So this thing that you see here, blue, it's basically people put in a promoter, okay, the DR5 promoter. Let's put the names here now, DR5. Is this thing working for you? This is not a blackboard. Are you guys kind of following me? Are you not confused? It's the first time I'm teaching in one of these rooms, so I'm confused. So don't be shy if you're confused. So you have DR5 as a promoter, and then you put GFP or GUS. In these days, people didn't have GFP, or it was not so common. We're talking about 1997. So what people did was putting GUS. So the more blue you have, the more hormones you have. So basically, this is like a normal picture of a growing seedling. You see that there's a lot of accumulation on the root, and then people flood them with this artificial auxin, and that's, what's your name? Yusuf predicted there's going to be a lot of blue, because basically the auxin went into the plant and basically activated this promoter, and you had a lot of production of this blue protein. Well, the protein is not blue, but produces a reaction that can be made blue. Okay? Cool, right? So now we have a tool to measure auxins. So now we can start kind of disentangling the fact that we see apparently pins going up and pins going down. So what's going on? I mean, is there still a gradient or a flux or whatever? What is it? I mean, and this is where things really started to be really interesting. Because when people start doing these things in roots, this is what they found. Huge accumulation of auxins on the tip of the root. And they were like, look at this. The whole plant looks like almost clear. And then you have like this blue ink dot on the tip of the root. So apparently it looks like, I mean, the whole plant was just producing auxins to send it to the tip of the root, which was, I would say, a completely unexpected result, because people were like thinking that there should be a gradient if the thing is transported from up to down or whatever. What the hell is going on? Moreover, I didn't spoke about it, but there's only one. There isn't just one root in plants, so I guess everybody knows that. But just that you make sure that you guys, we're all on the same page. So you have the main root, but when the root comes from a certain size, I mean, you start having what we call lateral roots coming out. Okay, so at a point when the root grows to a certain dimension, you start having lateral roots coming out from the main root. And people start thinking, so what about those? I mean, are those associated with accumulations of auxin as well? And this was the first demonstration that actually accumulations of auxins produce new organs. Now this is remarkable, because now, see, this cell here, it's a specific cell on the endodermis, and it's, look at this, look at around it. It's the only one that accumulates auxins. It's just one cell accumulates auxins. And this is enough for this cell to start dividing and producing a lateral root. This was the first time actually in animals and plants that anyone could pinpoint the accumulation of a certain molecule produces the de-differentiation of that cell into an organ. Okay? Which is something in that in animals it's almost like impossible, because if that happens you have cancer, but in plants it occurs naturally. Yes?
I have two questions.
Sure.
How did they generate this figure if they have to kill the legs, and how do they monitor it?
Yeah, well, I mean, we use tricks. So we kill, I mean, basically in a root you have a root which is little and a little bigger and a little bigger. So if you go along the root, it's like if you have a recapitulation of that. So they were lucky, of course, they got the right cell here, and one which is dividing a little bit up here, and the one which is already bulging a little bit up here. Or they looked at a number of roots and built a story, like in the documentaries of National Geographic.
Also, in the previous slide, you showed a picture where it was just at the tip.
Yeah.
So is this at the root tip, or is
No, this is lateral. This look at this diagram. This is lateral. So this happens here, not at a root tip. Okay, here's the thing. And this should be set to gas or to all stainings of that material. The more incubated, the more signal you have. So say like if you leave this material 1 hour with a reagent, you get a signal. If you leave it 2 hours, you get more signal. If you leave it like a day, you get a lot of signal. Okay? So you can just compare results if you use exactly the same incubation time. So my guessing is that one of these cells is somewhere around here, but you don't see it because there's just, not enough magnification, or basically because they just made a small exposure to see, oh, there's a big accumulation on the tip, but that doesn't mean that you don't have other smaller accumulations along the plant. Okay? Sorry, this is, sometimes there's tricks on these images, but that's a very good question, and this is the answer. Okay? Okay, so let's go back to the coleoptiles. We started with Darwin and the coleoptiles. Can this be applied to the coleoptiles also? And again, yes, finally. 100 years, well, more than 100 years, 110 years after Darwin made that observation, it is true. I mean, auxins go to the sites that elongates. So, and that was perfectly logical because we knew auxins induce cell elongation as well. Not just differentiation, but also cell elongation, depending on the concentrations. So this was a cool observation, and people actually could finally say, yeah, well, Fenton, Darwin, and all these guys were correct. So light is perceived on the tip, and then produces auxin, and because there's an accumulation of auxins on that side, those cells elongate more, and therefore the coleoptile bends. Okay? Cool. 110 years to explain this. This is just a slide to reiterate that. what about the root gravitropism? And somebody already spoke about gravity. Okay, you, I think. No, somebody. You, well, whatever. What happens during the gravitropic response? And this is when things get complicated, because it's not just the accumulation of hormone, it's the way in which the message is transduced into an action. So, follow me. In the coleoptile, accumulation of auxins actually induces elongation, and that's why the thing bends into this direction. If this was applicable to the root, say if you have a root like that, and you put it like that, and you know that it's going to bend, where should auxins accumulate? Up or down? Think about the coleoptile. Still up. Observation. It's the other way around. So in other words, it's definitely using auxins because they're accumulated on the downward side, but they're used in a different way. Okay. It can just be a matter of concentration. We don't know. Again, this just reports us that there's an accumulation of auxin. It doesn't tell you how much auxin is there. Or it might just be that these cells are adapted to respond differently than the cells from the coleoptile, which makes some sense because up there they're looking for light, down there they're looking for gravity. Okay? I'm just, but in both cases, by using these tools, by using this very clever approach of finding what is the receptor, what is the responsive element, what is the promoter, and putting a probe after the promoter, you can say where auxins are accumulated. Okay? And definitely, basically put the finger on the fact that wherever you have accumulations of auxins, something's going to happen. Either organs are going to show up, or organs are going to grow differently. Okay? Okay, so let's make a sum up of where people got. We're talking about the end of the 90s, beginning of the 2000s, so this is the turn of the millennium. And this is the kind of image that you guys are to be faced on papers if you want to read something about it, or on the textbooks. And usually, usually that you have two things that people try to make kind of correlations. One is the localization of the efflux carriers, the pins, which are here in red. And this is an embryo in two different stages. So you can see that even in the early embryo, the cells already are polarizing the pins. And the DR5, in this case with GFP, being accumulated on the basal part. So this is a very early embryo. It's an embryo with about 20, 30 cells. It's still round, but you can see that there's already a polarization and an apical and the basal part to this embryo. So the root, without surprise, is going to be developed from these cells, and the apex is going to be developed on this part of the embryo. Okay. The blue here is just it's a chemical that we use to stain DNA, and it's just for you to know that this is the cell nucleus. So for every blue spot that you have here, you have a cell. It's just for you to count the cells. Okay? Can you kind of synchronize yourself in this kind of images? Because I'm going to show you a lot in the next few minutes and next class. Okay? So auxins with one color, the pins with another color, because it's going to get really, really nice. It's one of the most beautiful stories in development of biology of any organism. Okay, so let's make a point of the situation. Now we can quantify auxins from tissues or cell extracts, but because there is expression of auxin in these genes, we can actually detect auxins by specific antibodies or by specific probes attached to artificial promoters. And secondly, by the orientation of the pin, we can infer the flux of the pins. Okay? Can we bring all these things together to make sense in why a root is a root, why a shoot is a shoot, and why everything should be in the right position? Well, okay. Let's think about the roots. As you said correctly, when you put the root, there's an accumulation on the wrong side. How come the auxins get there? Well, we have to think about these things. Now, we have a way of following where auxins are accumulated, and we have a family of proteins that apparently allows them to get there. So if I change a root from here like to here, and I see that there's accumulation of auxins on the basal side, how did the auxins get there?
The receptors change the rotation.
The receptors?
Or not, I'm sorry, the transporters.
The transporters, excellent answer. That's exactly what people found, but not on pin 1 or pin 2, on pin 3. Okay, so this is what you get when you change the route from being vertical to be horizontal. Pin 3 changes its location and basically redirects auxins for the bottom. And this introduces a very important concept, the localization of the transport that can be changed during development, during growth. In other words, the cell has mechanisms to read its context and change the positioning of the pins so that it redirects the auxins to different positions. And therefore, in this case here, there are cells which are specialized in sensing gravity. They're located about here. And what these cells are doing is they're signaling the cells surrounding it, change the position of your pins to send auxins to the other side so that the root bends. But that creates a problem. So you have this massive accumulation of auxins on the tip. I mean, like, how come they are accumulated there? Is there a way by which, all these pins are kind of inserted in the system to create these huge accumulations of auxins that tell, and what is the biological meaning of that? Why would a root have such a big accumulation of auxins in that specific spot? Well, of course, people start using pins and DR5s in all sorts of organs, and as you can see in the movie from Arabidopsis, and people have done this in many organs, people start seeing that there were accumulation of auxins not only in the tip of the root, but actually in every spot that's going to give rise to a new leaf or a new organ. In other words, it started to emerge a paradigm in which new organs are formed where you have accumulations of auxins. In other words, auxins are the trigger to say, oh, let's do a leaf. Oh, let's do a... by the way, I mean, in animals, it's the same thing. You have accumulation of certain molecules where you want to have a harm, and then a harm develops. Same thing happens in the fingers. There's accumulation of a certain molecule in the fingers, and fingers show up. Okay, and you can actually make, frogs with 7 fingers. If you pick up a bead and you kind of soak it with this protein and put it in several spots, and you have extra numerary fingers. Actually, we can be born with 6 fingers, and that's probably a mistake like that during development. Anyway, this is what you find. You have maxima of auxin accumulation in the points in which organs are going to be formed. And whenever you look at the pins, you see that there are specific patterns of pins that seem to be driving the accumulation of auxins in these specific spots. In other words, the pins are oriented in the sense that it looks that they're accumulating auxins in these specific spots, and that's why you have the development of new organs. So this started to be cool because we have kind of a very simple explanation how organs get formed and how cells interact and signal each other to tell, oh, you no longer are epidermis cells, let's do a meristem. Okay, let's do something new. And of course, in animals, this is also pretty much the paradigm. So now we're gonna study a specific case study that I think kind of boils it down more than, I know that the video you saw was all based on flowers, but that's just because Elliot Marvitz is a geneticist of flowering. He likes flowers. So everything which is like in the dirt, it doesn't care about. But roots are actually a much better system to understand this because it's more simple, and for anatomical reasons we can reach conclusions much faster. So this is how it goes. We're gonna analyze this game here. Stem cell niche maintenance in the meristem and pattern formation in roots. What? Okay, this story was pretty much, this is Elliot Marvitz. I mean, you saw it in a movie like 20 years before. This is a picture taken in a course I used to organize in Portugal. And the other guy that it's important for this story is this. I mean, everybody had a lot of alcohol, so don't make judgments. And he was wearing a mustache in those days. I don't know why he cut it, because it was kind of cute with the mustache. But this guy here is the guy that did most of the stuff I'm going to tell you about. I mean, there was more competing teams, so it's not just from him. I mean, it came from various teams, but because he's my friend, I'm prejudiced. They're both from my friends. Okay, let's talk about stem cells. What are stem cells? Who can tell me what is a stem cell? Where do we have stem cells in our body? In the bone marrow, okay? Not the bones proper, but the bone marrow, okay? Where else? In the?
Well, not in
Well, parts of the embryos have stem cells, not all embryos, but more. as adults. What is the other stem cell niche we have?
In the retina.
In
Retina.
the retina. Yeah, that's true. There's a small niche there. Also on the nose, we also have some stem cells. But you know, the canonical ones, the big ones, the one which really are relevant. It's the bone marrow and another one. Which one is it? Blood comes from the bone marrow, so there's not. You don't have, no. Okay, come on, guys. The testes, women, the ovaries. Okay. By definition, what is a stem cell? One. Secondly, which is in the continuous lineage of embryonic tissues. Okay? Everybody agree with me on that? And that's why, I mean, cells in the bone marrow never cease to, I mean, they never differentiated ever since the embryo. They always continue to produce blood cells. Okay? Same thing in the testes. I mean, since the embryo, they continue to produce spermatozoids, and in the case of women, they continue to produce ovules and to mature ovules. So there's a continuous lineage from the embryo until the adult person. Okay? Not in plants. Why? Because if we see a predator, we run. If an Arabidopsis plant sees a cow, it cannot run or it cannot cry. I mean, the only fate is that inevitably it will be hidden. Okay? So how did plants adapted to this kind of sad fate? They adapted by developing new organs. In other words, I mean, there's this thing called totipotency in plants that any cell at any point of the development can develop into a new organ. And this is just an adaptation thing. Otherwise, it would be a dead body after the cow went on, and we know that's not the case. They continue to grow. So, in other words, although there are small niches in the plant that actually comes directly from the embryo, most of the stem cell niches in the plant are actually generated by redifferentiation. In other words, cells differentiate and then they differentiate to redifferentiate, which in our case doesn't happen. Or when it happens, usually it's just the first step, which is de-differentiation, and that's bad news because you have cancers. Okay, basically, in other words, what cancers are in the beginning is you have a cell which is a liver cell, and that cell for some reason decides, oh, I'm not going to be a liver cell anymore, so it returns to a de-differentiated state, but because it lacks the coordination, it starts just dividing and growing and gives rise to cancer. Plants have cancers, but cancers are adaptive in plants. A plant does not, I mean, it's not a cancer in the sense of killing the plant, it's an adaptation, say, to a wound or something like that. So where do they reside? Basically, pretty much, you have stem cell niches at the tip here, which produce the new organs and the apical growth. You have stem cell niches here at the tip of the root that produces growth, a new root. And then you have stem cell niches that enlarge the plant. Okay? And then along the shoot and along the root, you continuously have stem cell niches that de-differentiate to produce new organs, new flowers, new leaves, new shoots, new roots. Okay? In other words, there are niches of cells that de-differentiate to become stem cell niches again and give rise to a new organ. So, question is, what do auxins have to do something with this? I mean, can we use the knowledge that we learned from this class, and now we got to auxin and pins, to understand how stem cell niches are defined, and do they give rise to new organs or to patterning inside the organs, which is a major biological question. How cells decide to be different inside the tissue, inside the organ. Okay? So keep thinking on that, and we'll talk.